What's the name of the second interpreter?
Right? It should be me. I don't know. We have used two the same. Let me just talk to him. Let's see that.
It's fine. I can see the two. If it's the two that have your
Yes.
name, it's fine.
Okay.
It's fine now, Washington.
All right. That's great. And I think...
Could you also tell me the presenters we have for today?
We have Prof. Han, then Prof. Shin, as well as Prof. John Kong. I'm not too sure if they've joined yet, but
Are those the names that they're joined with?
So, are you able to assist with identification of our presenters today?
Yes. So, what do you need? I think they are all signed in now.
I need their names so that I can assign them as speakers.
Mm-hmm. Can you see
the names that they are used to join on the webinar?
Okay, let me ask them.
Okay, thank you.
We sort all this. Please tell your colleagues that the session is about to start, so that
I think they are struggling to change their name. Otherwise, I can ask them to maybe we can raise their hands.
Yes, I have found Jihun Shin. It's fine.
Okay. And otherwise, I can ask them to leave some chat or raise their hands so you can find them easily.
Yes. That would be good. Let them raise their hands and I'll find them.
Okay.
Thank you.
right. Thank you. I think we can begin now. Good morning once again. My name is Washington, coordinating the AMRA GMP inspections. Today is our day 2 of the Q3 AMRA GMP training session. Yesterday, we started on a good note, and it was quite an impactful day with presentations from our colleagues from CEPI, Solomon. I think what a presentation he delivered yesterday. I think it enlightened us to the work that is being done by CEPI around the globe, and the great efforts in terms of vaccine development that are going on around. We are very grateful for that, Solomon and the CEPI team. We also had quite an interactive session, Q&A session, emanating from Solomon's presentation, and quite some great responses as well. We have since sent Solomon and the CEPI team the write-up of the responses. When that is ready, we will share with everyone during the course of the week. Let me start by introducing some of my colleagues that are joined today. There is Sue, who is a very, very big partner from the Africa CDC. She has been very instrumental in ensuring that this Q3 session is very successful. I think it's been almost 3 months us planning this Q3 session, and Sue has been pivotal in this role. So if you can unmute and introduce yourself, over to you.
Okay, thank you so much, Washington, and also good morning and good afternoon. It's my great pleasure to welcome you all to this virtual workshop on GMP inspections organized by AMRH and also as a sisters organization, Africa CDC. We are very appreciate all the efforts from the member states also to join this webinar. As Washington introduced me briefly, my name is Sugyong Kim. Everybody call me Sue. I'm a regulatory program lead at PHHM. So previously it's PABM. So PHHM refers to Platform for Harmonized African Health Products Manufacturing. So we now expand our scope beyond the vaccine, including all the medicines and also diagnostics. Also, I would like to extend my heartfelt gratitude to all the participants, speakers, and organizers who have worked tirelessly to make this event possible. Your commitment to improving the quality and safety of medicine in Africa is really commendable. And GMP inspection is one of the key, actually, towards the fostering the African health products manufacturing ecosystem. So I wish this opportunity and the workshop can give us fruitful insight and also the knowledge so inspectors can improve and also can make all the vaccine manufacturing and also some other products advanced technologies manufacturing possible in Korea in Africa, enhancing all the African population can access to the essential medicines shortly, I wish. And again, thank for all your dedication and participation. I look forward to productive and insightful workshop. Thank you.
Right. Thank you, Sue, for the introduction. I would also want to acknowledge the presence of my colleague, Julia, who has been quite very instrumental in supporting this. Julia, if you want to introduce yourself, please. Julia, if you're online, just
Yes.
pass yes.
Sorry. My name is Julia Kiguru from the AUDNA Secretariat and under the AMRH program, and I support the GMP TC. It's good to have you on board for this training. Thank you so much.
Thank you, Julia. And lastly, I think I want to acknowledge the presence of the GMPTC members that are joined at the moment. I see online there is Dominic and Mari. Dominic and Mari, if you would want to meet the team. I don't think they have a... Do they have speaking rights, Julia?
No, but I can...
No.
Let me just search for their names and allow them to talk.
Yes, and I hope there are also no other GMPTC members. If you are, you can just raise your hand and we give you the platform. All right. Then in absentia, I also want to acknowledge our GMPTC chair, Wayne Muller. He's quite busy this week, but every time he's checking on us and giving us guidance. And through his leadership, we've managed to come this far, and still there is a lot to be done. And we are glad to have him as our team leader. And also Alex is busy in Nigeria now. I see a lot of NAFDAC colleagues. If you please be kind to our leader, Alex. He's at NAFDAC this week, and he may join in at some point. Do take him out for some jollof rice there. And then Clarietta, who is also leading the RCD technical committee. I'm not sure if Cla is there. If you are there, Cla, you can also have a chance to just meet the team since this will be your team. Is Claretta in today, Julia?
No.
It's not joined. All right. No, that's fine. Did you manage to get hold of Dominic?
Yes, Washington and team, good morning, colleagues. I was trying to unmute. I hope everyone is okay. I'm Dominic Kariuki from Pharmacy and Poisons Board, Kenya, a member of the GMP TC. Thank you all, and thank you, the organizers of this training. Thank you, Washington.
Thank you, Dominic. And Mari? Madam Mari, if you are online, you can just also meet your team. Okay, I'm not too sure if she is able to do that at the moment, but nevertheless, let us proceed. So today we are going to have 3 presentations from Kenibit. The first one being the introduction from the chairperson, Prof. Han. Then we'll have a vaccine manufacturing bioprocessing, a general overview from Professor Shin. And then the third presentation is going to be on vaccine manufacturing bioprocessing, touching also like fermentation from Prof. John Kong, all these from University of Yonsei. And then we have our Q&A session. But before that, let me just give a brief recap from yesterday's session. Let's see here. So yesterday, we started with our pre-training quiz. And it's a very interesting statistics that we got from our pre-training quiz that there was evidence that surely there is need for us to conduct this training. So I'm happy that we have a big team that is lined up from CEPI, Kenibit, and tomorrow USFDA as well, so that they can fill in the gap that is shown by these statistics. So we have an average scoring of around 52% with a median of 47%, and it is our hope that post this training that we can move the median to well above the third quartile, as well as the average above the 50% or 75% mark. So, ladies and gentlemen, I think this is a good motivation for you to stay glued onto the screen, so that the presenters can fully impart their knowledge, so that you can understand the principles that you may have missed in the pre-training test, so that we can all improve our competencies. All right, and it was also great to know that our training sessions are getting popular. By each and every quarter, they are getting popular. If you could see that we had 444 respondents participating in yesterday's session. This is a remarkable statistic by any measure across the continent, and I've also seen across even at a global level. Having 444 participants in a training session is amazing. We thank you guys. Keep on telling your colleagues, spread the message, spread the link to your fellow inspectors, spread the link to your fellow assessors in the evaluation departments, so that they can also benefit from this initiative. All right. So my next recap session was on the CEPI presentations, where we had quite some insightful presentation about the institution CEPI, what they are doing across the globe, and where we also fit in as African member states into what they are doing. And it was great to know that there is a lot of work being done to prevent the devastating effects of pandemics that might come in future out of this, and we look forward to greater cooperation and support from CEPI, and also that There has been some questions, follow-up questions to the presentations, to the test, I mean, that was delivered yesterday. So I just want to give you a snippet of how spread the responses were and how maybe many of you missed these responses. You are going to get most of this the answers to these questions during the trainings that are happening. But if you would see that what is the primary function of vaccines, I think most of you, the answer was B, you got it right. And then what is one of the main challenges in developing vaccines for variable pathogens like HIV? The correct answer there was C, and 90% of you got that one right. And what is the primary purpose of using cell culture in vaccines? The correct answer there was to grow and propagate viruses, which is B, and most of you, 84%, got it right there. And then what is the key activity involved in downstream processing of vaccine development? It is my hope that the sessions are going to clearly they elaborate what is downstream, what is upstream processing. And the correct answer there was B, which is purification and formulation. And then if we go to which of the following statements is not true about mRNA vaccine development, I think this dovetails with a lot of questions that were being posed at Solomon yesterday about the fears around mRNA vaccines. But it is not true to say that the mRNA vaccine is integrated into the human genome, which was the main cause of concerns and fears and the like. Choose the unit operation that is not included in upstream bioprocessing. The correct answer there was the viral clearance. And again, this is going to be fully elaborated during the training. Choose the component that is generally not included in mammalian cell culture media. That is definitely IPTG. And we're going to also hear from the presenters what it is this. One of the comments was that, can you elaborate on the abbreviations? But I think through these trainings, this is going to be the abbreviations of our sector, the abbreviations of our industry, which we need to familiarize ourselves with as inspectors as we go to inspect these biopharmaceutical manufacturers. Choose the strategy among the following that is not used for scale-up in upstream processes. That is, the answer there is the constant agitation speed, and I think we're also going to get this from the presentation. Choose a host cell among the following for protein expression that is cost-effective, high-yielding, and suitable for producing small peptides like insulin. That is definitely E. coli. And the CHO cells are most commonly used host cells that produce the biopharmaceutical proteins. Choose the wrong one as an advantage of using CHO proteins as host cells, and that will be there is no genetic instability. That is not quite true. What is not purpose of sterile filter integrity? This answer is checking filter performance. All these other ones are quite also very correct. Choose the incorrect guideline on protein purification. The correct answer there was 2, which is maximizing number of steps. It is beneficial to know and to maximize the number of steps. So what are the 2 most important parameters in controlling TFF? And the correct answer there was number 4, which is cross-flow recirculation flow rate. And then which of the following statements regarding interpretation of column packing tests is incorrect? And the correct answer was the height equivalent to theoretical plates indicates the, I don't know what indicates what here. Then if we move to 15, which of the following is not appropriate route of administration for vaccines? That is ocular, number 4. Most of you got that one correct. Which one is of the following is not an appropriate component of vaccine for intramuscular injection? that would be a glidant. A glidant, this is for tableting formulations. And which of the following is not related to the chemical stability of protein therapeutics? And the correct answer there is precipitation. Which one is not correct with respect to manufacturing process of protein therapeutics? And the correct answer there was protect patient instead of the operator. We always want to protect both the patient as well as the operators working in these biopharmaceutical processing plants. In the freeze drying process, which mechanism or reaction is involved to remove water during the primary drying? And the correct answer is sublimation. So this is just a snippet of what was expected in the pre-test quiz. And the presenters are going to elaborate more on these correct answers that I've just been presenting. And I encourage you to sit back, take your notebooks, write some notes, and prepare for the amazing presentations that are in store from us. All right, so that is enough on the recap. Then I want to, without wasting time, I want to move on to introduce our very first speaker for the day, Prof. Han, who is the chairperson of KNIBIT, a very well-decorated academic. And I'm very sure that the entire KNIBIT team is very blessed to have him as the chairperson. Prof. Han is many academic achievements from PhD in chemistry from the University of Pennsylvania in the US, MSc in medicinal chemistry from Seoul National University, BSc in pharmacy from Seoul National University's College of Pharmacy, and he has worked at various institutions, mostly at Yonsei University, amongst many. His CV is just amazing. I would just want to stop here and give him the chance and opportunity to introduce himself and some of the works that he has done. Prof. Han, it is good to meet you again, Prof.
Okay, thank you, Washington. Thank you so much for your kind introduction for me. I think it is great to see you on our African colleagues. My name is Kyuni Han, and as the Washington introduced myself, so I'm the professor of the Yonsei University and since 2024, and I'm just working in chairperson as a KNIBIT. I think it's great to have you guys have the online the workshop, and today the two professors from our university are going to present, and upstream and downstream the process of the vaccine and the biopharmaceutical processing right now. So to save some time, I'd like to introduce my center. Starting with, yeah. We'll see. Okay. Let's see. Okay, I'd like to introduce the KNIBIT. KNIBIT is the Korean government supported training center. So we are doing right now the pilot operation period. So official training is going to start in 2025 with the two new buildings. So our program actually transferred from the Ireland NIBIT, which is the very well-known bioprocessing training program located in Dublin in Ireland. And so our training program is mainly focusing on the four modules, such as right here, the fermentation, which is upstream, and the purification, which is downstream, and the quality control, and as a final product. Today, the four professors from our center are going to present the fermentation and some of the the basics. And tomorrow, two professors are going to present the downstream process, which is purification of the protein and also fill and finish. And so far, 2024, since And we actually started our training program, and the actual hands-on training started in 2022, and those 13 units for domestic training in Korea, and two domestic the international training 2022, and then numbers has been expanded to the more than 711 for domestic training, and then 34 training for international training. Okay. So our training programs are basically fermentation, purification, and bioanalysis, and fill and finishes. So we have more than 300 hands-on equipment so offered two different types of the training program, which is mRNA vaccine biomanufacturing program, starting with the E. coli, the gene transfection, and then fermentation and isolation of genetic products, and the stabilization of the chemical process, and another the purification process, and the final fill and finish. And our actually the most important and the most well-known the training program is therapeutic antibody manufacturing training. The training program actually came from Irish NIBRT, and we actually working on the membrane cell culture system, and then an isolated protein from the cell, and we can identify the product and the bioanalysis and fill and finish it. And for the global engagements, and since 2022, so when we have the very severe, the COVID-19 pandemic season, and the WHO people visited in our center, so we arranged it in the first mRNA global vaccine biomanufacturing program, and supported by the Asian Development Bank. That training happens the 2022 to 2023, and particularly last year. So we actually finished 30 batches of the ADB training, and it had 3 weeks of online training for directive training, and then 5 weeks training for hands-on training. And this year, and we are doing the mRNA vaccine, the bioprocessing training program, and supported by the Right Foundation. It has been started 2024 May. So now the 41 trainees from 17 countries actually came to Korea, and they are in the second weeks of the training right now. And I'd like to mention that, okay, the last year training and the most training from the Asian Pacific region. And this year, so training region has been expanded to African region, to European country. So far we have among the 41 trainees, that we have the 21 trainees from Africa. So I hope you guys and to see you in Korea for your hands-on training sometime. Okay, I think this is all I have. Thank you. Over Washington.
Thank you, Prof. Han. What amazing work you are doing at KNIBIT there. And colleagues, you have heard the challenge that it will be great for you to also embark on some of these training programs that are happening at a global scale. Yesterday, during Solomon's presentation, someone was asking about PhD studies. Those are also opportunities that can be explored to improve the capacity amongst member states to regulate biopharmaceutical products. So I hope the colleagues are getting the challenge very well. Thank you very much, Prof.
Mm-hmm. Yeah, thank you.
All right. I want to move on to introduce the next presenter. Our next presenter is Prof. Shin, also from Yonsei. Prof. Shin is also a very well-decorated colleague from Yonsei, who's currently the assistant professor and chair of the Bioconvergence Major and Vice Chairperson of External Affairs at KNIBIT. He has worked in various positions and portfolio, starting with the National Cancer Institute in 2012, and in 2022 he was at Yale University as a postdoctoral associate, and then also at July 2023 he was at Yale as well as an assistant professor, adjunct assistant professor, and then at present he's at Yonsei as an assistant professor. He has done a lot of work in the biopharmaceutical space, and we are very glad and delighted that he has taken time to share with us his experiences and knowledge today. Prof. Shen, can you meet the audience, please?
Hello, thank you so much for your warm welcome. And let me share my screen here. Okay, minimize my screen. Okay, today, or thank you for your kind introduction. And I'm also very honored to be involved in this AMR RH GMP training. So today I'd like to introduce about the general overview in vaccine manufacturing bioprocessing. Okay. So here we have two contents that I'm gonna talk about. First, vaccine introduction, and then it will be followed by the overview of vaccine manufacturing process. So first, introduction for vaccine. To talk about the vaccine, I think we need to understand about the immune system. So here, the first part will be mainly focused on to understand what is the immune system and how immune response can occur upon the infection or against the so-called pathogen. So here is the definition of the immune response. Immune response is the reaction of the cells and fluid of the body to the presence of a substance which is not recognized as a constituent of the body itself. So good thing about our immune system is immune system can distinguish between our self versus non-self. So self here means that all of our host cells express the protein called major histocompatibility complex molecule, MH class molecules. All of our host cells express MH class 1 molecules, and all of our host proteins are presented as a small peptide, which is called an antigen on the MH class 1 molecules. Then our immune system can recognize all of our host-derived proteins or molecule as cell. However, when it comes to the like external, like a foreign, when foreign substance came into our body, which is our immune system has not yet been experienced, then those pathogenic antigens are recognized as non-self. So then our immune system starts to react against this kind of non-self and foreign substances. So reaction against those kind of foreign substances by our immune system is the immune response. Okay, then let's talk about the common sense, what's in blood. So we all know that in the blood, there are red blood cells, platelets, and plasma, and white blood cells. White blood cells are consist of leukocytes and lymphocytes. So today I'm gonna introduce about what might be the types of immune cells first to learn about the background knowledge in how to develop vaccine eventually. So leukocyte, let me first talk about the leukocyte first. One of the good examples is macrophage. Here you will see one macrophage is engulfing other cells by phagocytosis. So the target is entrapped by the macrophages, and then it basically engulfs it and then digests it. So this is called as phagocytosis. So this is the picture of the macrophage, and this red road is, let's say it's a bacteria. So when macrophage recognize the presence of foreign substances, such as a virus or bacteria, then they sense it and then they try to eat it, engulf it and digest it. that is called as phagocytosis. So here, phagocytosis of macrophage, when macrophage meet the pathogen, and then pathogen engulf the target pathogen by endocytosis, entrapment, and then it's gonna form the phagosome inside of the macrophages. And then this phagosome will be eventually merged with the so-called lysosome, another vesicle containing specific enzymes such as lysozyme. Then those lysozyme and then other enzymes start to destruct the engulfed pathogens such as bacteria. And then those digested bacteria will be exocytosed or also digested inside the cells. So this is the whole process of phagocytosis. And important function of macrophage here is that firstly sense the presence of the foreign body or foreign substances, and then immediately react as an innate immune response in order to get rid of the target pathogens. So that is the first role of macrophage. And then the question here would be how macrophage can sense the presence of foreign substances. That is, there are specific pattern of the pathogen, and then macrophage, those kind of innate immune cells, express so-called pattern recognition receptors. So, for example, bacteria, gram-negative bacteria, their bacterial wall is encapsulated by the coated by the lipopolysaccharide, which our host cell do not have. So when macrophage sense the presence of LPS, then macrophages are activated and then perform the phagocytosis in order to get rid of the target bacteria. So that is the first role of macrophage. And the second role is once the digestion happens, then a macrophage chopped up the engulfed bacteria. And so the chopped bacteria, for example, that chopped proteins will be, that will be, let's say, small, let's say that changed as a small sized peptide, which is also called as antigen. So those small peptides will be loaded on the MHC molecules, which I mentioned in the previous slide. Then macrophage will present the presence of foreign-derived antigen or peptide to other types of immune cells to cause adaptive immunity. So there are two important roles. One is phagocytosis mediated the innate immune response against the bacteria or virus. And the second role is to further activate the immune system. Macrophage present the antigen to other immune cell, which is T cell, I'll show you later, which is delivery of the pathogen information to other immune cells. And second type of immune cell is dendritic cells. Dendritic cell, as shown in this animation, you will see lots of dendrite. So using the dendrite, dendritic cells can capture target bacteria or virus, and dendritic cells are kind of a very professional cell for antigen presentation. Using their dendrite, capture the pathogen, digest it, and present their information, including pathogenic antigen, to T cell as an antigen presentation. So those are so-called as leukocyte and innate immune cells. When it comes to immune cell, again, that as a common sense, we all know that immune cells are derived from the bone marrow stem cells, which is called as hematopoietic stem cells. And hematopoietic stem cell will differentiate into two types of cells. One is lymphoid progenitor, and the other is myeloid progenitor. From the myeloid progenitor, it will differentiate into red blood cells or those kind of innate immune cells, including macrophages and dendritic cells. And from the lymphoid progenitor, it's going to differentiate into the B cell, T cell, and NK cell. And here, today, I'm going to introduce about B cells and T cells. So B cells is named by Bolsa of Fabricius, where the B cell firstly observed. And those are three important functions of B cells. Number one is antibody production. Second, memory response. And third, antigen presentation. So I think now we are getting to more familiar term, which is antibody here. So B cell, they do have a B cell receptor. when B cell receptors are bound on the B cell membrane, those are they are called as B cell receptor. Once B cells are activated, then part of them will differentiate into the short-lived plasma cells, and then they start to secrete their B cell receptor, membrane B cell receptor. And those of secreted form of B cell receptor, now they are called as antibody. So in this way, once B cells are activated, they produce high levels of antibodies and then secrete it. And part of them, so let's say if majority of B cells are differentiated into the plasma cell for antibody production, a small number of B cells will differentiate into the memory type of B cells to prepare the following infection or immune response for later. So those memory type B cells live longer and stay in our body. And once the second or third infection occurs, then those memory B cells immediately respond to the presence of pathogen to produce high levels of antibody directly. So those two are main function of B cell. And then lastly, so antigen presentation. When it comes to antigen presentation, all of our host cell, any cell express MHC class 1 molecules. And using that molecule, all of our host cell can present antigen to T cell. But in that case, that is through the MHC class 1 molecule. MHC class 1 molecules can engage with the specific type of T cell, which is cytotoxic T cell. I'm going to tell you later. And then there is the second type of MHC class molecule, which is MHC class 2. And then there are mainly there are, let's say, only 3 type of immune cells express MHC class 2, which are macrophages, dendritic cell, and here B cells. And then through the MHC class 2, this is the only way that can present the antigen to the helper type of T cells. So the lot of B cells that antigen, they are also known as professional antigen presenting cell, because macrophages, dendritic cell, and B cell, they can use either MHC class 1 molecules for cytotoxic T cell response, and then MHC class 2 molecule for the humoral or helper T cell response. That's why I firstly introduced macrophage, dendritic cell, and here B cells. And T cells, not like B cells, T cell, the T means to indicate the thymus, where the T cell matures. So there are another important type of immune cell, which is T cell shown in here. And then T cell also have specific type of T cell receptor. If I make a long story short, one T cell clone expresses one type of T cell receptor, and one type of T cell receptor can recognize, let's say, a single type of antigen. And in our body, we do have 10 to the 18 different types of T cell clone in our body. That in that way, with this kind of enormous numbers of T cell, our immune system can handle with uncountable numbers of external or foreign substances. And the major roles of T cells are like this. There are 2 types of T cell, as I just mentioned. Number 1 is cytotoxic T lymphocyte, also known as CD8 positive T cell, because they express CD8 molecule. And helper T cell, which are CD4 positive T cell or CD4 T cell, they do express CD4 molecule on their membrane surface. And like B cell, T cell also has memory response. So here, once T cell is, this is, let's say, before the activation, once T cell is activated by the T cell receptor signaling, then you'll see this is the nucleus, and then you can see the enlarged cytoplasm as about 2 times larger. That because once T cells are activated, then T cells start to produce lots of cytokines or immune-related molecule. That's why lots of transcription and translation will occur inside of T cell, enlarging the size of T cell like this. So those 4 immune cells are, let's say, I think, important immune cell type I'd like to introduce first to learn about the vaccine. And here that those are the most of the immune cell type in our body. And we can separate that as innate immune response and adaptive immune response. So innate immunity, which is the rapid immune response, that our innate immune cell can recognize the pattern of the pathogen and immediately responds in order to eliminate the target foreign substances. So I just used the image of policeman that capturing the thief immediately. But in case innate immunity is not enough to clean up the infected substances, then we need to activate the more powerful immune system, which is adaptive immunity. So this is rapid response, but adaptive immunity is slow response because it takes time to induce the adaptive immune response. So here, to induce adaptive immune response, the link is antigen presentation, which means the delivery of the information from the pathogen to the more powerful immune cell types such as B cells, which are the antibody secreting cells, and T cells which can directly kill the pathogen or pathogen infected host cells. And we already learned the types of antigen presenting cells. First, macrophage or dendritic cells, and B cells. So once those innate immune cells are activated or B cells are activated, then they will perform the antigen presentation to T cells. So using the MHC molecule, MH class 1 interact with the CD8 molecule. That's why through the MHC1 that those antigen presenting cells can stimulate CD8 T cells. And so MH class 1 is expressed in all of our host cells. So not only those 3 cells, other cells also can stimulate CD8 T cell too, if the condition is condition met the situation to activate the CD8 T cell. However, when it comes to CD4 T cells, only those of 3 cells, which are the cell type that express MH class 2, they can activate CD4 T cells through MH class 2 and CD4 molecule. Then once T cells are activated, CD8 positive cytotoxic T cell can directly kill the target cells. And CD4 T cell, which are the helper cells, they secrete lots of cytokines to further enhance other immune cell reactivity, such as stimulating B cell to secrete antibodies or activating macrophage to enhance their phagocytic reactivity against foreign pathogens. So this is the overall general overview of the immune response. Okay, so using this animation, let me briefly summarize about the immune response. So our body also has a physical barrier such as skin. So using this kind of physical barrier in the normal condition, bacteria or virus cannot infect our body. But upon tissue damage, they can invade our body. Then innate immune cells such as macrophage or neutrophil, other innate immune cells immediately respond to get rid of them. And part of them, dendritic cell, capture the antigen and then trying to do the antigen presentation to T cell. So through the circulation system, they will get into the lymphatic vessel and then get into the nearby lymph node, which is called a draining lymph node, to activate T cell, and T cell activate B cell to secrete antibody. And activated T cell and antibodies are going back to the damaged tissue area. Then now activated T cell can further enhance the reactivity of innate immune cell to completely clean up the infected pathogens. Antibody also capture the pathogen as well. Then in this way that the infection is resolved. So this is the overall way of immune response. And while it's not shown in this animation, in case of CD8 positive T cell, which are the cytotoxic T lymphocyte, they can directly sense the presence of infected host cell and then kill them. So, so far I've talked about the general process of the immune response. It is very important to understand the whole of those situations because then now we can think about how to develop a vaccine. Then to develop vaccine, we also need to further understand about the underlying mechanisms, how T cell will be activated. Because as is shown in the previous slide, once T cells are activated by antigen presentation, then part of those T cells, particularly helper T cell, will stimulate B cells and produce antibody. And also part of T cell will remain as a memory type of T cell for the second infection. So let me talk about the mechanism, how T cell can be activated. There are three signals for T cell activation. So here is the antigen presenting cells, and this is the antigen loaded MH class II molecules. So again, professional antigen presenting cells, macrophage, dendritic cells, and B cells are major cellular source of MH class II mediated antigen presentation. Then, if some T cell clone has an appropriate T cell receptor, which can bind to the peptide loaded MH class II, then it's gonna give the number of the first activation signals. And as you see in here, CD4 directly bind on the MH class II. That's why MH class II can activate CD4 T cell, and CD8 bind to the MH class I. That's why MH class I can activate CD8 cytotoxic T cell. So this is the signal one. And the second signal is so-called as co-stimulation. Once antigen presenting cells are activated, then they enhance the surface level of those B7.1 and 0.2 molecules, then providing this co-stimulatory ligand to the co-stimulatory receptor CD28 on T cell, it's gonna provide survival and proliferation signals. And then third signal is differentiation signal. Depending on the inflammatory conditions or situation, different types of cytokines are secreted, and depending on the environment, T cell differentiation will be determined by the mixture of the cytokines. And so those three are important signal, and signal 1 and 2 is pretty much enough to fully activate T cell. And here the important thing is the presence of second signal. If signal 1 is only present in the absence of signal number 2, then it can rather inactivate T cell, which means think about this kind of situation. If newborn baby will be exposed like high levels of such kind of beneficial protein in the milk, in the breast milk, then our body and our immune system recognize that as beneficial substances. In that sense, in that case, our immune system decide that do not react against this kind of beneficial substance. How our immune system can distinguish between beneficial substances versus harmful substances is whether the inflammation occurs. So once inflammation occurs, for example, viral infection happens or bacterial infection happens, then those of antigen presenting cells are activated, and then they will provide high levels of co-stimulation. So signal 1 plus co-stimulation will fully activate T cell and then followed by immune response. And this is the reason why when we develop vaccine basically is a
Excuse
kind
me.
of
Sorry, sir. Let me just interrupt you for 1 minute.
okay.
The interpreter was asking that you slow down a bit. You are too fast for them. They
Oh, I
wanted
see.
to be able to interpret for the French speakers. Thank you so much.
Oh, thank you for letting me know. I see. I'm gonna a little bit slow down because I was thinking that this might be very there are just too many things to talk about, but this might be one of the key information to understand the background that how we can develop vaccine. So in vaccine that we also put the adjuvant, and the role of adjuvant is to mimic the inflammatory condition. Without the adjuvant, if we are gonna just inject let's say viral protein, then our immune system may confuse that this might be a beneficial one. Then T cells against those viral protein will be inactivated and let's say hibernate. That is called as T cell energy or T cell tolerance. So if we want to develop proper immune response, then we also need to provide the environment as an inflammatory condition. That's why we provide adjuvant to induce co-stimulated signal number 2, together with signal number 1. Then we can successfully develop vaccine, like vaccine or new vaccines. Okay, then let me move on to the next slide. Then what is vaccine? Vaccine is a substance used to stimulate immunity to a particular infectious disease or pathogen, typically prepared from an inactivated or weakened form of causative agent or from its constituent or product. And this is a slide shows the impact of vaccination on selected diseases in the UK. This is the reason why we want to develop vaccines. So there are many different types of diseases, including diphtheria or meningococcus, poliovirus, or influenza type B, measles, and pertussis. So, for example, here, after the introduction of vaccination against diphtheria in 1940s, that the risk of the diphtheria is dropped like this. And that was the same thing for other types of disease as shown in here. So development of vaccination, as we also went through the pandemic, that vaccinating is a key for our like community and national health. Then how to generate the vaccine? So this is the picture showing the generation of an immune response by vaccine. So here we can inject vaccine. In this case, through the intramuscular injection, we can inject vaccine, which consists of vaccine antigen, the small peptide, together with adjuvant containing danger signal, which means that mimicking the inflammatory condition. So adjuvant will be recognized by the pathogen pattern recognition receptors, in this case by dendritic cells. And dendritic cells also like engulf vaccine antigen and then start to present those vaccine antigen through the MH class 2 molecule. So here is the peptide loaded MH class 2, or this is the peptide loaded MH class 1. So as I mentioned before, MH class 1 can directly interact with the surface molecule CD8, and then MH class 1 loaded antigen can stimulate CD8 T cells. Another name is cytotoxic T cells. Then part of them will differentiate into the effector T cell. Then those effector T cells will kill the target, let's say virus infected cells. And part of them will remain as CD8 positive memory T cells for preparing the following additional infection. And when it comes to the CD4 T cell activation, that peptide is loaded on the MH class 2 molecule. And those helper T cell, CD4 T cell, then they can further stimulate B cells. Then B cell will proliferate and differentiate into the short-lived plasma cell, producing antibodies like this. And few number of B cell will remain as a memory B cell for the memory response. So this is the overall process of immune response by vaccination. Then, so let me just add one more thing in this slide, that let's think about that why it might be difficult to design proper vaccine in some cases, such as, for example, COVID-19, that initially alpha, beta, gamma strain of the COVID-19 was the majority of the COVID-19 virus. But later, we saw that Omicron or other mutant strains were generated. Because of the rapid mutation of virus or bacteria, then that means that those kind of antigens can be changed. So through the pandemic, we learned that the spike protein of COVID-19 virus can be mutated. And the thing is, for example, if antibodies are generated against small peptide sequence of the spike protein, if that sequence has been changed, then previous antibody no longer able to bind on the target and then get rid of that. That's why, because of the mutation, that new virus strains can escape or evade from the immune response. That's why it is somewhat difficult to develop the permanent kind of vaccine against such type of virus or pathogens. Okay, and in this graph, we can see why vaccination is also beneficial. Here, from the first infection, that infectious agent, such as virus or bacteria, will proliferate. And after that, in our immune system, T cell will be activated, and then we can see the increased number of T cell after the infection. And once we have enough number of T cells, then antibody titers will be increased as well. Then with the activated T cells and antibody, infectious agent will be eliminated. And then after the resolution, that T cell will be decreased like this, while the antibody titers can be maintained like this. And after the first infection, if the second infection happened, then since there are already prepared antibodies and memory types of T cells, the second immune response is robust and very active. So immediately respond to the second infection and then do not provide the time for the proliferation of infectious agent. So very quickly, from the second infection, infectious agent will be cleaned up, and then the T cell will be decreased again, and then we can also observe the elevated level of antibody titers from the multiple infection. So using this kind of very basic concept of immune response, if we are gonna apply the vaccination instead of having first infection, then we can minimize the pain of the host, whereas all the T cell activation and then antibody production can be also generated by vaccination. So in this way, we can prepare our host for any kind of infection. So this is the principle of adaptive immune response following vaccination. And there are multiple different types of vaccines. If I summarize the types of vaccine using this slide, so there are live attenuated vaccines, weakened or inactivated, which was first introduced in 1798 against smallpox virus. And then this is licensed for other diseases such as measles, mumps, or rotavirus, BCG, etc. So basically that we can inject or we can immunize people with live attenuated, which means that we can reduce the amount of the virus and then inject it to host, or we can also weaken the, let's say, kind of dangerous part of the virus, and then using that as a live attenuated vaccine. And other type is killed whole organism or toxoid. Toxoid is also weakened toxin from some kind of pathogen such as diphtheria or tetanus. And also there are subunit vaccine. Subunit vaccine means that, for example, if we are going to generate the subunit vaccine against COVID-19 virus, then we can take out the spike protein and then make it as a subunit vaccine. Subunit vaccines are purified proteins or recombinant proteins, or sometimes polysaccharide and peptide. And also there are virus-like particles against human papillomavirus, and outer membrane vesicles, the vesicles containing gram-negative bacterial outer membrane plus pathogen antigen like this for group B meningococcal, and protein polysaccharide conjugate, the conjugation between polysaccharide and carrier protein for immunization. and viral vectors. So this is one of the very highlighted types of vaccine these days, that we can deliver pathogen genes or pathogen protein using viral vector. Not only Ebola, that nowadays we can utilize viral vectors for many different types of virus or bacteria, and on top of that, we can also use this for the cancer vaccine. And nucleic acid vaccine, now we are familiar with mRNA-based vaccine. This is nucleic acid vaccine. So RNA or DNA can be packed in the that lipid nanoparticle or lipid coat, and then it can be used for the immunization. And bacterial vectors, pathogen genes are in the bacterial vector. And also as a cell therapy, we can also use the antigen presenting cells. So those are types of vaccine. And lastly, in the first part, the herd immunity is an important feature of vaccine-induced protection. So here, diseased peoples are labeled as red, and then susceptible peoples are light blue, and then yellow means vaccinated people. When there is no vaccination, a small number of these people can spread spread virus or bacteria to all other susceptible people in the community like this. And if the vaccine coverage is below the threshold level for herd protection, then while the lead cannot spread to the vaccinated people, but it can spread into the susceptible people, and then the number of infected or diseased people will increase just like this. However, if the vaccine coverage is above the threshold level for herd protection, then even though there are few numbers of people who are still unvaccinated, but they can be protected by the herd immunity. So this is another important concept. We need to understand why we have to develop, or we need to develop, vaccine for various diseases. Okay, that was about my first part. And then let's move on to the second part, vaccine manufacturing process. So first, let me remind you about the central dogma. How do cells make proteins? So basically, we all have all the information about the protein is in the DNA. So by the transcription, mRNA will be generated, and then translation of mRNA will synthesize polypeptides, and then translational, post-translational modification eventually form the protein. So this is how any kind of proteins can be synthesized in the cell. So today, I'm gonna focus on more likely for viral vector or mRNA vaccine, which is lipid nanoparticle coated captured vaccine. So let me first talk about the viral vector. So a viral vector is a form of a virus used to deliver genetic material into cells. To use virus as a viral vector for genetic delivery, that researchers deleted the harmful factors in the virus, and then only used the leftover part for the delivery of the genetic element. So this can be used for any kind of vaccine against bacteria or virus. Also, not only with that, but also we can use viral vector for cancer vaccine as well. And the examples of viral vectors are adenovirus or adeno-associated virus, retrovirus, lentivirus, and herpes simplex viruses. And there are 5 key properties of viral vectors. So the first one is safety. As I just mentioned, in order to utilize a viral vector for genetic element delivery, that harmful components are already deleted. So in that way, we can safely use the viral vector. However, still there is another ongoing safety concern depending on the type of viral vectors. So for example, retrovirus or lentivirus, those virus when they deliver the genetic element, the delivered genetic element can be randomly incorporated into the host chromosomal DNA. In that sense, that after that, random incorporation of foreign DNA into the host chromosomal DNA can cause unwanted random mutation in the host DNA. So it can cause insertional mutagenesis, and if that mutation affect on the, for example, tumor suppressor genes or other essential genetic component, then they can cause harmful outcomes. That's why we also still need to consider about the safety issue when we use the viral vectors. And the second property is low toxicity, that also still this is basically derived from the virus. So, and also when we use viral vector, we do not want to see the excessive immune response from the host. So regulation of the proper level of inflammation is another key we need to consider when we utilize the viral vectors. And third, stability. Since viral vectors deliver genetic element, the stability of the delivered component is very important. So if some virus shows the unstable features for maintaining genetic element, then those types of viruses are not a good candidate for use for the development of viral vectors. So stability for the genetic element is another key property, and cell type specificity. So most viral vectors are engineered to infect as a wide range of cell type as possible, but depending on the types, that is also called as serotype. I believe later you're going to learn about from the other lectures that a virus has a serotype, and then serotype means that there is a specific type of proteins are there on the surface of the virus. So depending on the surface protein, such as spike protein in the COVID-19 virus, it decides what type of host cell will be infected. So cell type specificity, that if we can modulate this kind of properties, then we can also control what kind of target cell will be, that what kind of target cell we can likely change the genetic element. And lastly, selection. So viral vectors should contain selectable markers such as resistance to a certain antibiotics. So in order to select out the viral vector from other unwanted component. So those 5 are key properties of viral vectors. And there are 2 gene therapy modalities. One is in vivo and the other is ex vivo. So if we develop viral vectors, then we can directly use those viral vector as a vaccine. So it can inject viral vector directly to the patient or to the host. And another way is we can also take out the patient cells, such as patient blood cells, and then culture the patient-derived blood cells, and then using the viral vector, we can modify the genetic element of the patient-derived cells, and then expand them, and then infuse that back into the host's body. This is called as adoptive cell transfer. And good example of this kind of modality is CAR T-cell therapy. So CAR T-cell is basically we could harvest immune cell T-cell from the patient, and then we can overexpress chimeric antigen receptor against the target using the viral vector or other kind of genetic engineering techniques, and then expand those cells and then infuse them. Then those infused cells will find their target and then try to get rid of the target. So there are those kind of 2 modality in viral vector related mediated therapy. And those 3 types are commonly and widely used viral vector types: retro or lentiviral vector, adenovirus, or adeno-associated virus. So let me briefly introduce about those widely used viral vectors when it comes to pathology, capacity, genome makeup, integrated, etc. So first thing is, so retro and lentivirus can cause multiple diseases. but we engineer them as a viral vector and then only use for the genetic element delivery. One example of retroviral antivirus is HIV, that human immunodeficiency virus, which cause AIDS, the disease that acquired immunodeficiency. And adenoviruses usually cause respiratory diseases, and compared to those two other viral vectors, the AAV, it does not show really that disease association in humans. So that's why AAV is relatively safer than other viral vectors. And when it comes to the capacity for genetic element delivery, that retroviral is up to 8 kilobase, and then adenovirus is up to 35 kilobase, and then AAV is much smaller than other types of virus, so it only carries up to 4.7 kilobase. And depending on the virus type, there are RNA-based virus or DNA-based virus. Retroviral NTR, single-stranded RNA virus, and then adeno is double-stranded DNA, and then AAV is single-stranded DNA. And then this is the important thing you need to remember, that retroviral lentivirus, their genetic element will be integrated into the host chromosomal DNA. That's why upon the genetic delivery or upon the lenti or retro infection, the length of expression is long-term or permanent, stable. However, if we want to use adenoviral gene is transiently change the host cells, so it's gonna continue only about several weeks, and it does not integrate into the host chromosomal DNA. So as time goes by, that delivered genetic element through the adenovirus will be degraded. And AAV, like adenovirus, most likely degraded and transient, but compared to the adenovirus, it's a little longer, up to several years. And sometimes there is a chance that AAV delivered genetic element can be integrated into the host chromosomal DNA. Okay, so now let me talk about the overall vaccine manufacturing process very briefly. So here is the first part, vaccine candidate, and then plasmid DNA or stable cell line generation. This part is also included in the upstream processing. So if we are gonna simply separate the vaccine manufacturing process as 4 different steps, it's gonna be upstream process, downstream process, formulation, and fill and finish. So in plasmid DNA or stable cell line production, what we are gonna do is gene expression optimization, and productivity and quality-based cell selection, and genetic stability test, and research cell bank preparation. So development of cell lines, that we can develop stable cell line, typically mammalian cells that produce proteins at high level if we want to make antibody or viral subunit vaccines. When it comes to viral vectors, that we can also generate stable cell lines that produce virus or viral vectors. So what we're going to do is vector selection and codon optimization in order to enhance gene expression without changing the amino acid sequence, and transfection for the cell line for making stable cell line, and then lastly, select cells that express well, have good cell viability and productivity, and produce a lot of protein per cells or virus per cells. Then we're gonna develop the cell bank after the selection of those of clones. So this is how we can develop cell lines. And in the upstream processing, it includes cell line development, cell banking, cell expansion production, and harvest. So this is the upstream process, and then upstream processing is concerned with growing or culturing the cells that produce the therapeutic proteins or viral vectors. So during the initiation of the initial part of the cell culture, we are gonna inoculate the smallest amount of the cells, and then we are gonna expand the cell culture using the bioreactor as a huge or large scale level. Then later, we're going to harvest the cells or harvest the proteins or viral vectors by centrifugation and filtration. And animal cell culture, I'm talking about the animal cell culture in the upstream process. And then animal cell culture is the technology to separate cells from biological tissue and culture in vitro. And it's a production of biological drugs using genetically modified animal cells, such as stable cell lines, and key technologies used in the high value added biopharmaceutical industry. And the majority of biological drugs are derived from the animal cells, for example, CHO cell line, Chinese hamster ovary cells, and various methods depending on the types of incubator and culture we need to consider. So why is culture process development necessary? To secure high productivity and good quality of the target protein through homeostatic process development. So there are two issues in cell culture technology. Number one is complexity of cellular nutrient requirement. For example, that addition of amino acid, vitamins, or etc. is that essential component, many other essential components. And the second issue is cellular damages due to the external forces. So to maximize and then optimize the productivity of the target proteins or viral vectors, that is the reason why we need to develop the optimized cell culture condition. And let me move on to the downstream processing. Downstream processing includes purification, concentration, and maintain functional structure of the drugs or viral vector in this case. So those three are purposes of downstream: remove impurities and contaminants from the process, and concentrate the product to a safe and therapeutic dosage, and maintain structural integrity of the product in order to maintain the function of the drug. And then those are the parental product class that purity, efficacy, biological activity, stability, pharmacokinetics, pharmacodynamics, toxicity, and immunogenicity. So if I summarize all of those, that in three, that those are efficacy, safety, and quality. And downstream processing encompasses all process steps from cell culture harvest to the final purified product. And DSP is cost-intensive process that they can account for up to 60% of production cost. And DSP development it tends to lag behind in terms of the process scale and product recovery. So DSP, downstream processing, is perceived within industry to be the main bottleneck in large-scale production of biopharmaceuticals. And those are the examples of downstream processing for monoclonal antibody or virus like particle or whole virus or viral vectors and mRNA. The important thing during the downstream processing, as I mentioned in the previous slide, it is about the purification, concentration, and filtration. So, for example, mRNA, that mRNA, we first generate the plasmid DNA, and then from the plasmid DNA, that do the chromatography for the selection of the plasmid DNA what we want, and then in vitro transcription, then we can generate mRNA, then additional chromatography, and then ultrafiltration and diafiltration, and then further processing, that enzymatic capping of the mRNA for the maturation, that mRNA will be the 5' capping for mRNA will be performed by using enzymes, and then additional chromatography to isolate 5' capped mRNA, and then encapsulation with the lipid nanoparticle. Then mRNA vaccine will be formed. So this is the downstream processing for the mRNA. And another example for the viral vector, then viral vector will be harvested, and then cell will be lysed. So cells are producing viral vectors, and then those cells will be lysed, and then nucleus will be created such as benzonates, that because once cells are lyzed, then DNA will cause kinds of the web, or that DNA will form huge size of the kind of structure that can make the which can be a kind of interrupting part for the sample preparation. So nucleosid treatment will be performed, and then like mRNA vaccine, downstream processing, ultrafiltration or diafiltration, and chromatography, that eventually viral vectors will be purified and then filtrated and then concentrated. Then final fill and finish will be followed. And then other cases will be, so if those are virus and then mRNA and then capsulated by lipid nanoparticle, so there are also pretty similar but somewhat different downstream processing will be performed for recombinant protein-based drugs, such as monoclonal antibody or virus-like particles. And here is the overview about the upstream and downstream for the viral vector bioprocessing. So sowing of the cell line and then inoculation and then expanding in the bioreactor, making in the larger scale. If we are going to use the stable cell line expressing viral vectors, then we can directly harvest the virus from there. Or if we are going to use the normal cell line, then we can transfect the cell to produce virus. So by the transfection, then when cell will start to produce virus. After that, after the transfection, then cell will lyse, and then benzonase will be created to get rid of the huge size of DNA compartment. And then in the downstream process, that virus will be purified, filtrated, and then concentrated here through the filtration or tangential flow filtration or capture or polishing chromatography, and then concentration. We can eventually isolate the pure viral vectors from the through the downstream processing, and then it's gonna go for the final sterile filtration and aseptic filling. So the last part of viral vector manufacturing process is formulation and fill and finish. So this is the overall process of the viral vaccine manufacturing process: cell culture and transfection, and then virus production, harvest, and clarification for the virus, purification, and then formulation. Then here again that we can add adjuvant or stabilizer, and then those of finally formed product will be filled, and then inspection, labeling, and packaging. So this is the whole process of the viral vaccine manufacturing. And then lastly, let me briefly talk about the mRNA vaccine process. Again, let me remind you about the central dogma that mRNA is transcribed from the DNA. And then if we do have mRNA, if we are gonna deliver the mRNA to the host cell, then host cells are able to produce protein. If this is COVID-19 spike protein, then our host cell will produce COVID-19 spike protein, and then our immune system will recognize that and then start to produce antibodies or spike protein reactive T cells. So this is the mechanism of mRNA vaccine. So for example, COVID-19 virus, we can get the information about the spike protein. So natural mRNA contains intron part and then exon part, and then by the mRNA splicing and then during the mRNA maturation process, all the intron part will be removed, and then 5' capping and then 3' polyadenylation will be followed. But in this case, when during the vaccine design and then vaccine generation, that using the complete DNA sequence, we can artificially generate complete form of the mRNA, and then this mRNA will be kept with the lipid nanoparticle like this. And if we are gonna deliver this lipid nanoparticle, which is the mRNA vaccine, through the injection, Then this lipid nanoparticle will be endocytosed, which means that engulfed by our human host cells. And then from this mRNA, that protein will be translated, and the human cell will start to express proteins such as spike protein. Then our immune system will be immunized against this spike protein. So there are so many advantages using mRNA vaccine. Non-infectious, long-term stability, rapid, reproducible, easily engineered, and large scalable production, US legislation, cytogenic delivery, induction of T and B cell responses, and rapid production synthesis with shorter half-life, egg and cell-free, does not interact with genome, and FDA does not consider non-replicating mRNA vaccine as gene therapy. So benefit of mRNA vaccine during the pandemic was designing and then generation of a new mRNA vaccine was very fast. That's how that for the COVID-19 that we could very actively and then efficiently develop the vaccine against the COVID-19 vaccine. And then there are those kind of disadvantages, such as few efficacy report and no long-term safety profile, including an activation of cellular immune sensors and rapid degradation of naked unmodified RNA and extracellular barriers and inefficient endosomal escape delivery vehicles needed, and small molecule drugs could impact mRNA metabolism. But anyway, if I just simplified, the advantages of mRNA vaccine is just simple and safe and quick to generate. And here is the overview of the mRNA vaccine manufacturing process. From the plasmid DNA and then using the bacterial system, we can easily produce huge amount of the plasmid DNA, and then we can purify it, and then using the plasmid DNA as a initial DNA, then we can do the in vitro transcription to generate the mRNA, and then mRNA will be kept, and then after that, that filtration or chromatography, and then removal of the contaminants such as human DNA, then those will be concentrated, filtrated, and then purified, and then eventually formulated by lipid nanoparticle. And then this will be go to the fill and finish, and then visual quality check and labeling packaging. So the different part of mRNA LNP vaccine is formulation and fill and finish. The formulation means that using various types of lipid, such as ionizable lipid or pegylated lipid, phospholipid, and cholesterol, that we can pack the already prepared nucleic acid, such as mRNA, and then form the lipid nanoparticle. So this can be eventually used as a mRNA-based vaccine or nucleic acid-based vaccine. And then those two are a widely used machine to generate the lipid nanoparticle. That Kane and I would also have those two instruments. And after that, lastly, fill and finish in this kind of environment with the barrier technology, and then E-beam, which is electron beam that decontamination tunnel, and then aseptic filling environment. And also, we could lyophilize the mRNA vaccine as well. So in this way, we can do fill and finish, and this is the last part of the mRNA vaccine production or bioprocessing. So thank you very much all for your thank you for your attention for the very long time. And then later, I'll be happy to take any questions.
Thank you very much, Professor. Wow. Wow. What a serenade of great pathophysiology, immunology. This indeed is a great recollection of what many of the inspectors learned in college. Thank you very much. Very detailed presentation, and I've seen a lot of requests from the chat that can we get this presentation, and I know you already shared. we
Yes.
will share with the team as well.
Okay.
I see if you have time to just pop in the chat box and you see how well appreciative people are.
I truly appreciate it as well. And then sorry that I was also considering about the time limit that I don't know. I was thinking that whether I'm gonna pass the time.
No, but you did very well. So colleagues, we will share this presentation. I think Julia, now that is finished, you can also start sharing it in the chat box, so that people can meditate upon what you were saying. I think you took us through the whole journey, through the baseline definitions, the development of the drug substance, right up until the fill and finish, everything. I think this was a great presentation. So, is there anyone with questions?
Okay, I was watching the Q&A chat window. That is there a factor to control cell reverse back to the pathogenicity at any stage, or what kind of a harsh condition lead the cell to reverse to virulence? Okay, so if I understand it correctly, so when it comes to the generation of viral vectors or mRNA vaccine, that the first thing is due to the safety issue that people that we need to get rid of all the virulence factors first. But still, that yes, whenever I give my lecture, that I always say that in biology anything is possible. So still, there are many ways that some cells can reverse back to the pathogenicity. But for example, that mutation. But in your question, so I need to think about whether when it comes to viral vector, but it's very less likely because the viral vector means that already that viral replication ability has been already removed. So we only use the viral vector, only use the produced viral vectors, and then those viral vector only transfer their genetic element into the target cells. Hopefully this might be an answer for your question. And I also thank you for your warm mention. I shared my slide, and then I want to know why we took booster vaccine for COVID-19. Was the first vaccine not able to produce enough T cell and antibody? And if so, why? So if you remember one of the slides I showed you, compared to the first infection or vaccination, the second infection always shows the higher levels of antibody titers. That's why through the multiple vaccination, we can maximize the potential of vaccine. And so that's why we usually took the booster shot. Not only COVID-19, other vaccines such as BCG or many other vaccines also usually suggest or recommend multiple, depending on the vaccine, that multiple shots. Well, there are many, so I don't know how much time I do have.
You can go ahead. I think we will stretch for 10 more minutes on the Q&A session.
Okay. Then a 35-year-old woman with a history of severe allergic reaction receives her first dose of an mRNA COVID-19 vaccine. Within 15 minutes of vaccination, she experiences the symptom of anaphylaxis, including hives, swelling, and difficulty breathing. What is the most likely cause of her reaction? Or this is a really difficult question because there are so many possibilities. I don't know whether so allergic reaction that to induce the allergic reaction, that usually it's gonna be triggered by allergens. For example, if somebody has a peanut allergy, that if peanut is ingested, then it's gonna show the allergic response. I personally do not think that COVID-19 vaccine contains allergen in this case, but still, like other vaccine, mRNA LNP also functions as an adjuvant, which can trigger the innate immune response. And since the response was very quick within 15 minutes, which means that it's gonna cause the very like immediate and rapid innate immune response, and then cause the shock such as anaphylaxis. So again, that yeah, I cannot say what is the direct cause, but yeah, there is a possibility that injection of something can cause the severe shock event, something like that. However, still we don't really understand, but there might be a kind of not the direct or what I mean is some biological cause. Rather, it can be also emotional response. If somebody is frightened by the vaccination or any kind of injection, that can also cause shock. Same thing can be happening in the allergic response as well. Surprisingly, for example, if somebody has a rose allergy, somebody are allergic to the flower rose, some of those allergic people, when they watch the picture of the rose, then they show the allergic response. So allergy is also kind of the response that also connected to our emotional or neuronal like response. So it is hard to say what is the direct, but yeah, there might be possibility that vaccination can trigger the kind of the response. And can you help me understand how multi-epitope based vaccines are produced? What is the advantage of such vaccines? So, depending on the size of antigen or peptide loaded on the vaccine, that can be, we can say also, where epitope means that usually when antibody recognize small, or like let's say about 9 to 20 short peptide, those are called as epitope. And also sometimes, since protein is a 3D structure, even though peptides are not directly connected to the peptide bond, by the location, the 3D structure can be recognized as a that epitope as well. So if, for example, if we're gonna generate a vaccine with the spike protein, that being said, it already has multi-epitopes. So having multi-epitope would be, I guess, beneficial because all of our human do not have we do have different immune system. So even though we are immunized with the same vaccine, the immune response will be different. I mentioned that average number of the T cell clone number in one person is 10 to the 18. But it does not necessarily means that all the T cells repertoire is the same among our different peoples. So because of that, having multiple epitope will more likely induce immune response in different people. So there might be advantages, but again, there is a limitation to deliver the amount of the genetic element or antigens. One example is, as I shown in the table explaining about different types of viral vectors. So we need to consider what might be the most efficient way to provide bacterial or viral antigen or epitope as a vaccine. And then what are the quality control technique for vaccine manufacturing process? I think this part will be answered by other lecture because my lecture is just overview. And please say something on vaccine with adjuvant and without it, and advantage and disadvantage. Oh wow. Okay, so vaccine with adjuvant. As I said, without adjuvant, if we are gonna inject a huge amount of antigen, that it's gonna tolerize our immune system. For example, this might be a kind of extreme case. Let's say if we are gonna inject the spike protein of the COVID-19 without the adjuvant in our body, then there is a chance that host T cell will be tolerized, and which means that they're gonna go for the hibernation. They no longer wake up, and then they do not react against COVID-19. Then that is not the case that we want to see, right? So in order to see the vaccination or like memory response, then adjuvant is necessary. But still, there are many techniques that replace how to replace the adjuvant, because the thing is we don't really completely understand the mechanism of adjuvant. There are like many adjuvants. Usually, what people use is a fluid adjuvant or incomplete fluid adjuvant or alum. So alum is basically the liquid form of the aluminum, and then we are using that as adjuvant, but we don't completely understand how they can act as an adjuvant. So these days, for example, mRNA vaccine, LNP, that LNP composition can replace the function of adjuvant. So we do not really need to put the adjuvant in that case, but there are multiple things, because as I mentioned previously, in some cases, we can, in the viral vector, we also add the adjuvant there. But anyway, so there are advantages and disadvantage of using adjuvant sometimes would be, since we do not completely understand the underlying mechanism of adjuvant. So if we can completely understand the mechanism and then we can control that would be the benefit. So still, depending on the modality that we can choose whether what type of adjuvant will be used, or we can replace adjuvant with others. And is there any possibility of the mRNA vaccine interacting with or impacting the host genome? So mRNA vaccine, okay, so in general, I can say no, mRNA vaccine will not interact with the host chromosomal DNA. But honestly say, in biology, anything can happen. So depending on the sequence or depending on the product, it can indirectly can affect host genome. It could be possible depending on what kind of mRNA you are gonna deliver. And during the manufacturing process of an mRNA vaccine, which step is critical to ensure the stability and efficacy of the final product? So during the mRNA vaccine, critical ensure the stability and efficacy of the final. I guess this part, I believe, also will be further covered by other more detailed lectures. But like initially that forming mRNA, and then also like good quality of the lipid nanoparticle formulation is a critical step. And also we need to completely remove the possible contaminants such as human DNA, because that is also another very harmful thing if that is contaminated. And what type of parameters will look very carefully when evaluating combination of vaccine at dossier evaluation and disadvantage of using combination of vaccine? What type parameters will look very carefully when evaluating combination of vaccine at dossier evaluation and disadvantage of using combination of vaccine? Well, frankly speaking, I cannot completely understand the question. Let me take a look, and then possibly, if possible, I'm gonna answer it later. And why are extractables and leachables more important for biologics? Well, there might be, I think, also this might be also the question that probably answered by other lecture. And is it possible that patient can react to adjuvant added to vaccine? In such case, how do the manufacturer control this? And is allergy caution necessary in the labeling requirement? I think the last part is supposed to be controlled by the medical doctor who's gonna directly provide the vaccine. And is it possible that patient can react to adjuvant added to vaccine? In such case, how do the manufacturer control this? So when it comes to adjuvant level, also the amount, the dosage of the vaccine, so that is always the very first thing. For example, even Moderna, they first tested, well, in that case, that was the mRNA vaccine. So as far as I know, there was no adjuvant, but they at least first checking the dosage of the optimal dose for the vaccination, and then decide depending on the age or depending on the well in the COVID-19 mRNA vaccine case, it only separates as for the adult or young children, but usually the vaccines that dosage and adjuvant level is supposed to be firstly tested and then decided. And why is E. coli preferred during fermentation? Okay.
Prof. Shin, I'm going to have to interrupt your flow a bit.
All right.
So what we're going to do is we have copied all the questions. Then we're going to share with you all the list of the questions that have come, and some are still coming in, so that maybe you can provide a written response which we can then give to the participants. But in the interest of time, I want to move on to the next presenter, if you allow me.
Thank you very much.
All right, Prof. Thank you very much. Wonderful session you have delivered for us.